All right, welcome back everyone for more nervous system physiology. So last time we talked about the membrane potential and how the opening and closing of ion channels influences changes to that membrane potential. So now let's talk about how changing the membrane potential is going to influence whether or not we get the action potential that we need to send messages by using the nervous system. So we've already said that as far as a graded potential goes, a graded potential is not an action potential. What we're going to see here is that the graded potential determines whether we get the action potential or not. Graded potentials come in two different varieties based on the way that we change the membrane potential. If the graded potential is a depolarization, meaning the inside of the cell becomes more positive, then we call that a EPSP, or an excitatory postsynaptic potential. So just as a little bit of review, what kind of an ion channel opening do you think would trigger an EPSP, sodium or potassium? So I'll let you kind of figure that out on your own, but on the other hand, if the graded potential instead causes hyperpolarization, in which the inside of the target cell becomes even more negative than it already was, we call that an inhibitory postsynaptic potential. Again, sodium or potassium, which one do you think opening would cause an IPSP? Well, regardless of what we get, we obviously know by now an EPSP is desirable if we want an action potential, but even if you do get an EPSP, you are not guaranteed to get an action potential. There's a few requirements that have to be met. So let's go ahead and get a better idea for what a graded potential actually is. A graded potential is a localized change in the membrane potential, meaning that the membrane potential of most of the cell is still at rest. It's just in a very one specific area that it has changed. But what we're going to have here is we want that graded potential to spread throughout the cell. So if you imagine that 99% of the cell is at the resting value of -70 and 1% is at, say, +30, what we're going to want here is those positives to spread throughout the remaining 99% of the cell. But the problem here is that the graded potential is going to get weaker the further that it spreads out, and that's kind of the issue here. So in most cases, not all, but in most cases, a graded potential is going to be caused by the opening of a ligand-gated ion channel, which is what you see here. This channel that you see here that allows sodium through is a ligand-gated sodium channel that was opened by the release of neurotransmitter by this axon terminal. That's what the stimulus is. So when we open this sodium channel, sodium is able to flood into the inside of this neuron here. We're looking at its cell body, and you can see kind of some of its dendrites on the side here. Sodium rushes in, and we produce a graded potential, what we would call an EPSP, an excitatory postsynaptic potential. So this region of the neuron here that you see shaded in red is now very much depolarized, but everything else that you see in blue here is still at the resting value of -70. But as we let more and more sodium ions in, those sodium ions have the capability to migrate through the cell body towards the other part of the region of the axon that you don't see illustrated down here. And as those sodium ions migrate, other parts of the neuron will become depolarized as well. But here's the issue. The strength of the graded potential will be the greatest at the point of entry of the ion channel of the sodium coming in. As the sodium charges start to spread out, the signal and the strength of the graded potential gets weaker and weaker. That's what you're looking at up here. At the origin point, meaning the point at which sodium entered the cell, the strength of the graded potential is as high as it's ever going to be. But as we move out from 1 to 2 to 3 to 4 to 5, the graded potential gets weaker and weaker and weaker and weaker. And as we're going to see, if we want to get a graded potential, if we want to get an action potential, the graded potential has to still have a certain amount of strength by the time it makes it to the axon hillock. So to summarize here, graded potentials are caused by ions moving through open ion channels. So the potential will be strongest at the point of entry, but as I said, because those ions migrate and their concentration becomes greatly diluted the further that they spread out, the graded potential becomes weaker and weaker. Okay, so let's get something straight. If a neuron is going to communicate over a very long distance, does the graded potential sound like something that we want? Something that gets weaker the further it has to travel? No, goodness no, right? So what we want is we want an action potential. And the nice thing about an action potential is that it will not lose signal strength no matter how far it has to travel. So it definitely seems better than the graded potential in that way. So all the graded potential is really here to do is to give us the action potential. Either give us the action potential or do not give us the action potential. So the graded potential, from the time that it enters into a neuron, it will trigger the action potential if those positive charges, if those sodium ions migrate from their point of entry to the axon hillock, what is also either called the trigger zone or the axon initial segment. This is the part of the neuron that we have to get the graded potential to get to at or above a certain strength. If we are not able to do that, we are not going to get an action potential. So if the ions do make it to the trigger zone, and if we can depolarize the trigger zone to at or above that certain threshold value that we talked about earlier, then we are guaranteed to get the action potential. But if our graded potential, if it's an EPSP, if it is too weak to depolarize the axon hillock to that particular membrane potential value, then we do not get the action potential. So it's for this reason that an action potential is sometimes called all or nothing. You either get the action potential at 100% strength, or you get absolutely nothing. There's nothing in between. So this is a very important slide here because we are actually going to look live at two different situations, one in which we do not get an action potential and one in which we do. Now, let's set a few things up here. So the big cell that you see right here is the cell that is going to receive the graded potential. This axon terminal that you see up here is the neuron that is signaling to this cell right here. So when this axon terminal releases neurotransmitter, that is going to open ligand-gated ion channels on this cell body, and that will allow something like sodium to come in, and that creates the EPSP. Okay, let's look at something else here. Do you notice how we have 3 different voltmeter electrodes stuck into this neuron at 3 different places? Those 3 different places are at the origin point where those sodium ions come in, and we understand this is where the graded potential will be its strongest. We have another one placed about halfway down the length of the cell body. We understand that as those ions migrate, the graded potential will have gotten weaker by this time. And then finally, we have a third electrode placed just outside the trigger zone. So we have to be at or above that threshold by the time the graded potential passes this electrode right here. Okay, one last piece of information before we jump into these 2 examples. Do you remember how I said that we have to reach a certain threshold? Well, for an action potential in a neuron, that threshold is negative 55 millivolts at the trigger zone. So if we're going to get an action potential, we have to satisfy 2 different requirements. The graded potential has to migrate to the trigger zone, and once it gets to the trigger zone, that graded potential has to depolarize the trigger zone to at least negative 55 or above. If we get to negative 56, we get no action potential. If we get to negative 55 or anything above it, we will get an action potential. Okay, so let's look at these 2 examples here. Let's start at the example on the left. So the neuron axon terminal up here releases neurotransmitter. We open a ligand-gated ion channel, and we get our EPSP here. So a bunch of sodium flows in, and those sodium ions are going to start migrating down the cell body towards the axon hillock, towards the trigger zone. If we look at our first electrode here, we see that the graded potential has depolarized this part of the cell body to about negative 40 millivolts. That is terrific. That's great. We are above threshold. But unfortunately, getting above threshold up here does not mean diddly squat. We need to get to threshold down here at the trigger zone, and we understand the graded potential is going to lose strength the further that it travels. So after it travels a bit and gets down to the second electrode halfway down the cell body, you can see that we have already lost considerable strength in the graded potential. We are down from negative 40 and we are down to negative 55. We're right at the threshold. So things are not looking good for us if we want an action potential, because this signal at negative 55 is going to get even weaker as we continue on down towards the trigger zone. And as you can see, right when we get to the trigger zone, the graded potential is now at about negative 60 millivolts, and that is below threshold, and since we did not meet the requirements, we are not going to get an action potential. The graded potential reached the trigger zone, but it did not depolarize the trigger zone to at least negative 55. And because of that, our interpretation is that the graded potential was too weak to justify firing off an action potential. Okay, so let's look at the example over here on the right. So same sort of thing, we get a graded potential, this time a stronger graded potential that gets us to, looks like about negative 35 or negative 30 millivolts. By the time we get down to halfway down the cell body, we are still comfortably above the threshold. We're at about negative 40 millivolts. And by the time that we get to the trigger zone, we are still above threshold. We're at about negative 50, negative 45 millivolts. So because the graded potential reached the trigger zone and we depolarized it above that threshold value right there at the trigger zone, that meets all the requirements, and this time we will fire an action potential. So whether you get an action potential or not, it all just boils down to that one very specific thing. Do you get to threshold or do you not? That's all that matters. Okay, so now let's actually start talking about the action potential itself. We know what you have to do to get an action potential. You have to have an EPSP to get you to threshold. Now we can start talking about the action potential itself, assuming that we met all the requirements. So in our discussion of the action potential, we're going to make all the proper assumptions that we got an EPSP and we got to threshold. So when threshold is met, this is going to trigger the opening of voltage-gated sodium channels on the axon hillock. Now, do you remember in our previous discussion when we said what opens a voltage-gated channel? A voltage-gated channel will open when the local membrane potential is at or above a specific value. So this should answer our question as to why is the threshold negative 55 millivolts. That's the number because that is the number that is necessary to open these voltage-gated sodium channels. These are the things that have to open to give us the action potential. Okay, so when we open these voltage-gated channels, which direction is sodium going to go? Is it going to enter the axon or is it going to leave the axon? Well, because there is a lot of sodium in the extracellular fluid, sodium is going to rush into the axon. And this will cause even further depolarization of the axon membrane, and it is going to drive the membrane potential even further up from negative 55 to negative 50, negative 40, and so on. We are going to continue to let sodium in and drive the membrane potential up until it gets to about positive 30 millivolts. At that point, these voltage-gated sodium channels will become inactivated, meaning they will no longer be capable of letting any further sodium in, which is why we peak at positive 30. You can see this here. If we start at negative 70 right here, we get our graded potential at number 2 right there. That gets us to threshold, at which case the voltage-gated sodium channels open. They let a whole bunch of sodium in, and that depolarizes us up until about positive 30, and then that's kind of where we are at this point. So at about the same time that sodium stops entering the cell, voltage-gated potassium channels are going to open up. So both of these types of channels exist on the axon membrane. The sodium channels open first, and just as those sodium channels inactivate, we open up these voltage-gated potassium channels. All right, so for potassium, which direction is potassium going to go? Is it going to enter the axon or is it going to leave the axon? Well, because potassium is very highly concentrated in the intracellular fluid, potassium is going to leave, meaning the intracellular fluid in the axon is going to become less positive and more negative, which is going to drive the membrane potential back down, back towards the negatives. This phenomenon is called repolarization, in which the membrane potential goes back towards its resting value. It is different than hyperpolarization. Hyperpolarization is a term we specifically use when we go below the resting value. If you're just going down, then it's repolarization. But if you go from negative 70 to negative 80 and so on, that is hyperpolarization. So this falling voltage portion here for number 4 represents potassium leaving the axon to go into the extracellular fluid, and it continues to do so until we get back to the resting membrane potential value. At least that's ideally how it should be. These potassium channels are very slow to close, meaning that we're going to let more potassium out than we really meant to. Meaning that instead of going straight to the resting membrane potential, we're actually going to hyperpolarize this cell a little bit. This is what's called overshoot. So we let more potassium out into the extracellular fluid than we really meant to, which is what accounts for us going below the resting value, and now we're down here. So in order to get ourselves back to the resting membrane potential value, we need to get some of that excessive potassium in the extracellular fluid back into the intracellular fluid. Can you think of a way that we can pump potassium from the ECF back into the ICF? So I'll let you kind of think about that. All right, so let's talk about a few things. So we went through that kind of quickly. Let's go back to those voltage-gated sodium channels. So any voltage-gated channel is going to have what's called a activation or voltage gate, meaning a little gate that will open and close depending on the value of the membrane potential in the area. So for this voltage-gated sodium channel, it's this little swinging thing that is attached to the plus-minus dealie here. So when we get to threshold, this gate is going to open, it swings open, and now sodium can rush into the cell, which gives us our depolarization phase. So the voltage gate will be open as long as the membrane potential is at or above negative 55. But sodium cannot pass through unless both the voltage gate, this one here, and another gate on the bottom here, which is denoted by this little swinging ball and chain called the inactivation gate, if either one of those gates is closed, sodium cannot move through. They both got to be open. So when it comes time to inactivate these voltage-gated sodium channels, when we get up around positive 30, the voltage gate will still be open because we are still comfortably above threshold. Rather, the inactivation gate swings around and blocks the bottom of the channel, which prevents any further sodium from getting in. So the voltage-gated sodium channel will stay like this until we fall back below threshold, at which point the voltage gate will close and the inactivation gate will open again, and we reset things so that we're ready for the next membrane potential. So that pretty much discusses everything that happens in one action potential. There's a lot of information there, a lot of stuff to try to wrap your head around. So what I recommend you do, take a little break, go over that again, and make sure that you understand everything that happens during one action potential. Everything from the time you depolarize the membrane to the threshold, to the opening of the voltage-gated sodium channels, to the opening of the voltage-gated potassium channels, until we get back to resting membrane potential value. Okay, so one thing that we probably need to clear up is that An action potential refers to the series of events that we just discussed happening at one very specific part of the axon membrane. If an axon is going to conduct a message completely down the length of the axon, that is going to require many, many, many action potentials happening one right after another. The imagery I like to invoke here is it's kind of like knocking down dominoes. So every action potential is a domino that needs to be knocked over, and in order to effectively transmit a long-distance message, we need many action potentials leading to other action potentials further down the line. So the first domino is just going to be the action potential that you get at the trigger zone. For reasons that we will discuss later when we talk about saltatory conduction, all you have to do is knock down that first domino, and then this action potential here will lead to an action potential right next door, and then that one will lead to number 2, and then number 3, and number 4. So each action potential will guarantee the initiation of the next action potential down the length of the axon. Like I said, it's kind of like playing around with dominoes. The whole fun of dominoes is that if you set them up in a row like this, you don't have to painstakingly use your fingers to knock each one of them down. All you have to do is, if you set them up correctly, all you have to do is knock down the first domino, and then each one will knock over the next one in the line. So again, this is why we say that action potentials are guaranteed. All you have to do is hit that threshold at the trigger zone, and you guarantee that the other action potentials are definitely going to happen. Okay, so let's talk a little bit more about action potentials and how the nervous system actually uses this to communicate messages. So as we said, the only thing that is actually required for the firing of an action potential is that we get to that threshold value of -55 at the trigger zone. So the action potential strength is going to be the same regardless of how far above threshold we were or whether we just barely got to threshold. So this is something that's important. There is no such thing as one action potential being stronger than another action potential. Every action potential that is fired by a neuron is going to be identical to every other action potential that that neuron fires. So what you're seeing here is you're seeing in this graph 3 different action potentials. We know that these action potentials are identical in terms of their intensity or strength because they all have the same amplitude, meaning how high does the voltage get before it comes back down. So each one of these gets up to about +30 as we saw, so they all have exactly the same strength. What you're seeing here more specifically with these red arrows is you are seeing a series of stimuli of increasing intensity as you go from left to right. The leftmost arrow is the weakest stimulus, and the rightmost arrow is the strongest stimulus. The weakest stimulus, as you can see here, produces no action potential. We can interpret that as it produced a graded potential that was not strong enough to get us to threshold, so we get no action potential. The next arrow appears to have just been strong enough to give us an action potential. But you'll notice that the third and fourth arrows, even though those are even stronger stimuli, they do not produce stronger action potential. So every action potential is going to be exactly the same. It doesn't matter how strong the stimulus is. Every single individual action potential will be identical to every other one. So the question then becomes, why are action potentials all or nothing? Why do we either get them at a specified value or why do we get nothing at all? So we've already covered the idea that if a graded potential is too weak, we will not open any of those voltage-gated sodium channels because the requirement for their opening is we have to get the axon hillock depolarized to -55. Okay. Well, now imagine that you just barely get to -55. Let's say you just barely get to -55, and you're only able to open a few of those voltage-gated sodium channels, maybe like 1% of all of them. Well, even if you get just a few of those sodium channels to open, that is going to produce more depolarization because you get some sodium flowing into the axon. That further depolarization will open more of those voltage-gated channels that you didn't open before. They will start letting sodium in, and you'll get this positive feedback loop occurring until 100% of the voltage-gated sodium channels are open. So this is why we say all you have to do is get to threshold. Even if you only open one of those channels, by getting it to open, you are going to guarantee that the rest open, and you will guarantee the completion of the action potential. Now, let's discuss why we cannot possibly get action potentials that differ from one another. So, an impulse of higher magnitude, meaning a stronger stimulus and a stronger graded potential, it cannot produce a more significant action potential, because we cannot get the amplitude any higher than positive 30 millivolts in the action potential graph. We know that this is the point at which we inactivate the voltage-gated sodium channels, and it's also the point that we open voltage-gated potassium channels to go back down for the repolarization phase. So we cannot get a stronger action potential because the amplitude for these reasons will literally not go any higher than positive 30. The only other way you could possibly interpret one action potential as being superior to another, if that were a thing, is maybe one action potential lasts longer. Well, that's not the case either, because every action potential will also last for exactly the same period of time, because everything is working on kind of a timer here, because you deal with voltage-gated sodium channels inactivating, and then they reactivate. So that is what determines how long the action potential occurs. Okay, but here's the deal, though. Don't read any of this just yet. I want you to do something for me. Don't go overboard here, but just take your skin and just kind of pinch it a little. So pinch it very lightly. Pinch it as lightly as you possibly can. You probably aren't feeling much of anything at all right now, right? Well, the idea there is that you're producing such a weak stimulus that the graded potential that you are forcing on those sensory neurons in your skin is probably not strong enough to give you an action potential. Okay, we've established that. Now start pinching a little bit harder. Continue to pinch harder and harder until you can really kind of feel some pressure on your skin. So what you're doing there is you're producing stronger and stronger and stronger graded potentials over time. So this is something we want to clear up here. Why is it, if every action potential is exactly the same, why is it that you could feel greater and greater and greater mounting pressure on your skin? How is your brain, how is your central nervous system able to interpret the stimulus getting stronger and stronger? So basically, the strength of the stimulus has to actually mean something, though, right? Well, here's the deal. Stimuli of varying magnitudes, whether they're very weak or very strong, will not produce different action potentials. We've already covered that. What they will do is produce more frequent action potentials. So here we're looking at the same sort of thing we were looking on a previous slide. On the left here, we have a very weak stimulus that we apply for a few seconds and then we take off. Then we come in with a stronger stimulus that we apply and then take off. And then finally the strongest stimulus that we apply and then take off. So on the far left here, if we look at the membrane potential graph up here, we're at resting membrane potential. As you can see here, the weakest stimulus does not produce any action potential. So again, that graded potential was too weak to get us to threshold. But look at what happens here with the medium strength stimulus. For the few seconds that medium strength stimulus is applied, we get 4 different action potentials. Okay, but you'll notice each action potential has the exact same amplitude. They're all identical. When we come in with the strongest stimulus yet, we get even more action potentials. If you compare individual action potentials to one another, they're all identical. But with the strongest stimulus, we get more and more and more action potentials fired through the axon in a given length of time. So this is how our central nervous system interprets the strength of a stimulus. It's not paying attention to what one action potential looks like compared to another. It's looking at how many action potentials are coming in a given length of time. So the reason why your brain knows that a hard pinch is different from a weak pinch is because with a hard pinch, more action potentials are coming through that axon as compared to a weak pinch. That is how your central nervous system is able to interpret the strength of a signal, whether it's how hot something is, how painful something is. The stronger the stimulus, the more action potentials are going to be conducted through the axon. All right, let's go ahead and wrap this video up here. So when we come back next time, we are going to start talking about refractory periods, basically an application of some of the stuff that we just got done talking about in terms of limiting how many action potentials can be fired by a neuron in a given length of time. So I look forward